Hi, welcome back. So last time we looked at internal factors and we saw that cells have many types of kinases and that they are always present inside the cell. And those kinases that help control the cell cycle are actually activated by cyclins. So cyclins are a group of proteins that are readily made. Cyclins are a group of proteins that are readily made. That are readily made and destroyed at certain points in the cell cycle. And destroyed at certain and destroyed at certain points in the cell cycle. So those are what we are calling cyclins. Cyclins are a group of proteins that are readily made and destroyed at certain point of the cell cycle. So these two factors, that is the external and internal factors, help a cell advance to different stages of the cell cycle when cell binds to each other. When cells binds to each other. The next thing that we are going to look at is that even though cells are meant to divide, some cells need to die out. And that is what we will be looking at. We will be looking at how the body actually organizes itself and destroys some of these cells. But let's go through a short video and look a review about the external and the internal factors. Then we see what is on here. So we already discussed here that cell cycle is controlled and there are checkpoints. We talked of cyclins. We said that cyclins are a group of proteins that are rapidly made and destroyed at a certain point in the cell cycle, and their main action is to activate kinesis. And we saw that a kinase is an enzyme that, when activated, transfers a phosphate group from one molecule to a specific target molecule. And we saw that the action here is to increase the energy of the target molecule and change its shape. So let's see how we can get the picture from here. We can get a picture from this. So we are having here cell cycle control and checkpoint activities. So we are expecting that as the cell divides, it will go through checkpoints. So this is protein kinase. So it goes from G1 up to the last here, and it will be stopped. So cyclin levels in G1, the level increases and increases and increases to the maximum, then this leads to a kinase to be active, and this might stop the continuation to the next level. Cyclin dependence kinase is just what we talked about here. We say that they depend. These kinases depend on cyclin level, and cyclin's level may be released at any point of the cell cycle. So here, if it drops, it will affect the kinase. It will affect the kinase. So we have almost 3 checkpoints. We have the first one after the G1 phase or stage. We have another one after the G2, and we have another one at the M phase. So a cell can actually cease to divide when it reaches when it is still at before it enters the S phase, due to either lack of enough energy and such stuff, or it may not progress to mitosis because maybe the DNA didn't duplicate, or maybe other organelles didn't duplicate, so that cell will be destroyed. Or actually it may enter mitosis and fail to enter to finish cell division, and that is cytoplasm. So we will be looking at these details later on. Right now, I just wanted to highlight it. So we will continue with our discussion. Now we will be looking at apoptosis. Now we will be looking at apoptosis. What actually apoptosis means is programmed cell death. Program cell death. So not all deaths are actually bad. Some deaths are programmed and they are good for the organism, and apoptosis is just one example of a good cell death. It occurs when internal or external signals activate genes that help produce self-destructive enzymes. So many questions remain about this process anyway, but what is known is that the nucleus of an apoptotic cell tend to shrink and break apart. The nucleus of an apoptotic cell, an apoptotic cell, the nucleus of an apoptotic cell tends to shrink, tends to shrink. After shrinking and break apart. So after breaking apart, of course it has been destroyed already or killed, and break apart. And the cell is recognized by the immune system, and the cell is recognized by, it will be recognized by cells in the immune system. Immune system. These cells, that is the cells from the immune system, very tidily globe up the apoptotic cell and recycle its chemical parts for use in the building of other molecules. So if this is the cell, they will global it up, which means they will destroy it or eat it up. After eating it up, then what we have here is actually the nutrients, and those nutrients will be used to build other molecules. The picture you see here shows example of apoptosis. So you can see here that these fingers here are actually webbed fingers. They are webbed fingers. So as the fetus grows, so in the early stages of development, human embryos have webbing between their fingers and toes or digits. But before the baby is born, those cells typically go through apoptosis. So they will go through apoptosis and therefore open up here so that we end up with those digits of fingers that babies like to put into their mouths, and the toes too. So that is what we mean by apoptosis. That is what we mean by apoptosis. Apoptosis actually, apoptosis is controlled by internal and external factors, just like the ones that control other cell growth. So external, we have intrinsic pathways which are inside. So this one can help destroy the damaged DNA. So it can help destroy the damaged DNA, or apart from damaged DNA, there might be release of some proteins that will basically destroy the whole cell, so that the cell will actually be completely destroyed. Or some intrinsic factors may attach, extrinsic ones, may attach on the cell wall and lead the inside of the cell to produce chemicals that, after attaching here, of course, we talked of active site. So if it attaches on that site, it leads others to attach here, and this leads to release of chemicals that bind with others and lead also to the programmed cell death, which we are calling actually apoptosis. So that is what we call apoptosis, programmed programmed cell death. So in cancer cells or in other cells, cell division is uncontrolled. In cancer cells, cell division is uncontrolled. In a normal cell, division is normally controlled. But as the cell becomes malignant, then it ceases to be controllable. So when we were looking at cells, we saw that cells go through cell cycles, and cell cycles are controlled. We also saw that we have programmed cell death, which is apoptosis. But what we want to see now is what we call cancer. So we are going to look at what we call cancer. So as the name goes, when you hear the name, it is actually scary because cancer is one of the leading causes of death. So cancer is the common name for classes of diseases characterized by uncontrolled cell division. Is a common name It is a common name for, sorry, I'm writing far instead of for, a class, for a class of diseases, of diseases characterized by uncontrolled cell division. Characterized by uncontrolled cell division. So, unlike other types of previous types of division, this is also a cell division, but the only difference is that this one is uncontrolled. This is uncontrolled. So because it's uncontrolled, it's a big deal. So it arises when regulation of the cell cycle breaks down. It arises when regulation of the cell cycle breaks down. When regulation of the cell cycle breaks down. So when regulation of the cell cycle breaks down, this will lead actually to what we are calling cancer. So unlike healthy cells, cancer cells grown in culture dishes continue to divide, even when surrounded by neighboring cells. Even when surrounded by neighboring cells. So cancer cells can also continue to divide in the absence of many of the growth factors required for division in healthy cells. Even if some things are lacking, these cells will still divide. Even with the release of chemicals to control cell division, they will still ignore the instructions, as we can put it, and continue dividing. So as a result, they divide much more than do the healthy cells. They divide much more. So we will stop here. In the next video, we will be looking at the details of what cancer is and how maybe it can be controlled.